Okay, today I want to share with you the solution for this test. Chapter 20, that's about photosynthesis. Okay, photosynthesis. And I will explain the questions one by one. I hope you see from the structure PQRST, and you can recognize structure first. You can recognize the structure. This is from your textbook, page 14. From your textbook, page 14. And first of all, you write down the name for PQRST first. Okay, P is P is the upper epidermis. Okay. Q R S T. Okay, for Q, we call this the palisade mesophyll. R is the spongy mesophyll. S is the vascular bundle. That should be the xylem and phloem together, which transports material. Okay, vascular bundle. Or T we call this the guard cell. Okay, first of all, you recognize all these first, and then this is page 14 of your textbook. And I want you to write down what's wrong, what's correct here. Which of the following correctly describes? So structure P is impermeable. This is A is wrong. Why is it wrong? Because it's not the structure P being impermeable to water. Okay, it's the cuticle. I write the correct answer behind. Okay, cuticle is impermeable to water. Okay, so you have to pay attention. Cuticle. So you have to pay attention that cuticle is different from upper epidermis. Cuticle is the layer on top. Upper epidermis is a layer of cells covering the top layer of cells or on that leaf. Okay, how about structure S? Structure S transports gases. That is not correct. Okay, structure S transports water and minerals and food and nutrients. Okay, S transport water and minerals and nutrients. So B is wrong. C. Structure T allows gases to diffuse in and out. It's not the structure T that allows. It's the stoma, not guard cell. Allows gases to diffuse in and out. Okay. So pay attention. Don't mix up stomata and guard cell. Structure T is pointing to the guard cell. So the last one left is QRT. QRT have chloroplast, okay? Mesophyll, guard cell. All three of them have guard cell. So this question, the answer is D. Okay. Question 2. Question 2 refers to refers to the conditions necessary for photosynthesis. Okay. Necessary factors for photosynthesis. What are the factors? You should be able to remember that it requires carbon dioxide, it requires water and light and chlorophyll. Okay. Let me see. Okay. This is on page 12 and before. Several setup from page 8 to page 10. Okay, page 8 to page 10. Which setup are used to investigate the need of for chlorophyll? So when you study the need for chlorophyll in photosynthesis, first of all, you set up one group. Okay, have chlorophyll versus not have chlorophyll. Chlorophyll. Plus all other necessary conditions. Okay, this is the condition that we need. That we need to compare. When you need to study the need for chlorophyll, okay, you need to have one setup that have chlorophyll and all other necessary conditions. For example, carbon dioxide, okay, water and light. Carbon dioxide, water and light. CO2, water, light. Okay, as a result, you should be able to compare the and set up P. You compare the white area and the green area only. Okay, you compare P only. Whereas in set up Q and R, then set up Q and R, they either lack CO2 or they lack light. So it's not providing the necessary conditions for your photosynthesis. Okay, so we do not compare to investigate the need of chlorophyll in photosynthesis. So the answer is A, question 2A. Okay, let's move it. Question 3. Which of the following correctly showing the the green area? The green area. Okay. As I said, we need all four of them. In the green area, we have chlorophyll. Okay, and set up R, no light. In setup Q, there is no carbon dioxide. You can see potassium hydroxide pellet is responsible for absorbing CO2. Okay, absorbing and removing CO2. So in setup Q, there is no carbon dioxide. In setup Q, there is no carbon dioxide. So setup Q and R cannot produce starch. Q and R set up Q and R cannot produce starch. So it gives brown color in iodine test. Okay, whereas in setup P, we have blue, black in color. Okay, so the answer is B. Last one, question 4. How do we speed up the investigation? For example, you can let's talk about question 2 and 3 first. The increase, this is statement 2, increase to 35 degrees. So enzyme becomes more active. Okay, so 2 is correct. Set up option 1 is wrong. When you supply green light, supply green light, it cannot be absorbed. All green light will be reflected. All green light is reflected. So supplying more green light does not help. Supplying more green light does not help with speeding up photosynthesis. Okay, and also in setup Q, all CO2 is removed, or you can say absorbed, and set up Q already. So in setup Q, there is no CO2. Putting more potassium hydroxide pellet does not help with speeding up photosynthesis. Okay, so Q is also wrong. As a result, we say B is correct. Okay, only number 2. And this question 5. Question 5, first of all, you have to recognize W, X, Y, Z. Recognize the structure first. Okay. And the structure, W, X, Y, Z. For W, we call this the thylakoid, or it's the thylakoid or the granum. For X is the stroma, for Y is the starch granule. For Z is the outer membrane. Okay, outer membrane is not anyhow useful. It's just going to wrap up everything in the chloroplast, just surround everything. Okay, so and according to page 24 of your textbook, in thylakoid there is, or you can say granum, there is photochemical reaction. Okay, in stroma there is Calvin cycle. Okay, so we want to say water is for the reaction taking place in W. For reaction taking place in at W, we do not need water to trigger the reaction. Okay, we do not need water to trigger the reaction. We need light to trigger the reaction. Okay, so A is wrong. Number 2, carbon dioxide acceptor, that is the carbon dioxide acceptor, that is the 5 carbon compound. 5 carbon compound. 5 carbon compound is located at X, located in stroma for Calvin cycle. This is correct. Okay. Third one, glucose is C. Glucose is produced at what? This is not correct. Okay. Glucose is stored at Y. Stored as starch granule. Okay. Glucose is stored at Y. And for Z does not help with capturing light energy. It's just the chloroplast outer membrane. It has no function, just to wrap everything. Okay, not as Z. If you want to make correction, if you want to do correction for this statement, you can say light is captured at W. Okay, so question 6. The question asks you, from W to X, we transport some materials from thylakoid to stroma. Okay, that means from this thylakoid to Calvin cycle. What is transported there? Let me draw again. From thylakoid, this stack of thylakoid, we call it granum, okay, to Calvin cycle. We transported something called ATP and NADPH. So as a result, ATP and NADPH. So the answer is 2, 3. This is D. Question 6. Okay, question 7. Some of you may want to take a look at teacher's notes. Okay, you can refer to this teacher's notes on page 24. Okay, we want to trace where the oxygen go. We want to trace where the oxygen go. So in this case, if you want to know Okay, for example, there is a carbon dioxide. Okay, CO CO2 carbon dioxide molecule. Okay, this oxygen is a special oxygen atom. If this oxygen is a radioactive atom, 放射性嘅原子 okay, where will the oxygen go? The oxygen must go into carbon cycle and must go into glucose. Okay, the oxygen will not be transported through NADP or ADP. Okay, so we will not transport it in this way. The oxygen must go into glucose. For the other, okay, water molecule, H2O. I use purple for this. I use pink for this oxygen. This oxygen atom comes from water. Water will be broken down and release oxygen. Okay, so this oxygen contains O16. O16 is the oxygen gas that you produce from photochemical reaction. For the glucose should contain O18. This is a special radioactive oxygen atom. Okay, carbon dioxide labeled with oxygen O18. Okay, so the answer is C. Okay, you should be able to trace where the oxygen goes. Okay. Next one. Which of the labeled stages require energy from ATP formed from photochemical reaction? So according to page 22, some stage require ATP. For example, regeneration of carbon dioxide acceptor and the reduction of 3C compound. Okay, from 3C to triose phosphate, we need ATP. From triose phosphate to 5-carbon compound, we need ATP. So the answer is A. What is molecule X? Molecule X is glucose, of course. How do we use glucose in this case? For glucose, it's used in this way according to page 28. Okay, page 28, what can you produce from glucose? You can produce a lot of materials from glucose. For example, it has an energy source for photosynthetic cells. Okay, for cells that is carrying out photosynthesis, you also need to carry out respiration. Okay, this is a raw materials for its respiration. Okay, providing energy. Second point, to synthesis, glycogen is wrong. We do not store glycogen. We will store starch. Okay, you should store starch instead. Third one, you also have cellulose. Cellulose is to be synthesized to make new cell wall. So 1 and 3 are correct. So you should choose B. Okay, last one. How to speed up the rate of photosynthesis? So you can refer to the greenhouse. Okay, this is page 35. Page 35. Okay. For example, on the cloudy and rainy days, you give more light. This is correct. Okay, more light energy. Supply more energy. Use a heater to increase the temperature. It's also correct to activate the enzymes. 1 and 3 are correct, but 2 is not correct. Okay. Well, no matter whether you use the glass to allow red light or green light, red light or blue light to pass through. Okay. No matter whether you use it or not. Let me see. Okay, here. I can show you. Red plus blue plus green versus red plus blue. Okay. So anyway, the amount of energy absorbed that can be absorbed is red light plus blue light only. Red light plus blue light only. So, and the right hand side, there is no green light. And the right hand side, there is no green light. So, the energy available for absorption. Yeah, the light energy available for absorption is the same for both sides. Okay, so you have same amount of energy available for absorption. So in this case, I can say statement 2 is wrong, so I will choose B. Okay. So for the part below, I have explained during the class. So I have just opened the file for including the answer here. So we want to, first of all, you have to study the purpose of this experiment. Okay, we want to study the effect of carbon dioxide concentration. Okay, when you want to study the effect of carbon dioxide concentration, you want to change concentration of sodium hydrogen carbonate solution. Okay, so it provide different amounts of CO2. You get what I mean? So we only want to change one factor. That is the only factor that I want to choose. I want to change is the amount of CO2 available. I want to keep everything constant. So in this case, I would say the thermometer is used to ensure the temperature is constant. Okay, I want to use that thermometer to keep the constant temperature. Okay, so that temperature would not affect. The rate of photosynthesis. Keep a constant water temperature. Okay, so that temperature would not be a factor to affect the rate of photosynthesis here. Okay. The only factor that affect the rate of photosynthesis is CO2 concentration. Okay, so for dependent part B, dependent variable. Dependent variable is talking about the amount, how much we measure at last. What is the factor that we measure after the experiment to show the rate of photosynthesis? Okay, you should measure. Okay, something to be measured to show the rate of photosynthesis. Okay, something to be measured. So as a result, we should measure the amount of oxygen produced. Okay. Part B2. What assumption was made when using the dependent variable to indicate the rate of photosynthesis? In this assumption, okay, just now we measured the amount of oxygen produced. Okay, we measure how much oxygen is produced. Right? Oxygen is produced and collected in the pipette. You collect some oxygen gas there. We have to assume that, okay, first point. Rate of respiration is more or less constant. The rate of respiration is constant throughout the experiment. Okay. The rate of photosynthesis, rate of respiration is constant throughout the experiment, so that when more CO2 is available, more oxygen is produced. When more CO2 is available, more oxygen is produced. So the oxygen produced would not be consumed in at a greater rate. Okay, so the amount of oxygen produced can truly reflect the rate of photosynthesis. Okay. So due to the time constraint, I can explain up to here. I'll hope to make another video, or I can show it to you during the class. Okay. And for question 11C onwards, I will make another video to you to tell you the answer later on.